Hi guys. By now, I don't know if you're familiar with the rules, but let me just get them out anyway. You have like 7 minutes to present your talk and 3 minutes for the discussion part. And please try to stick to this 7-minute talk session, and all the questions will be asked during the 3-minute discussion part. So apart from that, I guess everything is all right. So Avital, you can start your talk.
Hello, everybody. Yeah. Can you see my presentation?
Yeah, we can see.
Okay, but it's not full screen, right? Yeah, now it is. Yeah. So my 7 minutes starts from now. So hello, everybody. My name is Avital Reisman. I am a master student under the supervision of Professor Ofer Manor in the colloid and surface phenomenon groups. So in our research, we're trying to understand the physical principle of the coating phenomenon, which is induced by surface acoustic waves. And as you can see on the right side, there is a video, and we can see a silicone oil that partially coats a water drop. The contact line of the silicone oil is the red line, and the contact line of the water drop is the blue line. And it moves under the, we induce the moving by the surface acoustic waves. Okay, let's talk why we need this coating effects. So nowadays there is a great interest in local and small scale manufacturing. So therefore, scalable and small coating systems are very important. And here we come. In addition, nations all across the globe raced to reach the moon and faraway planets, right? So one of the challenges is exploring space is manufacturing durable spacecrafts and robots. Currently, the contact industry is far below to fulfill this need. That is why all these 5 elements, we should receive it. So I think that coating systems supposed to work in low gravity environment. In addition, it's obvious it must be durable, right? And also small features, small features in order to manufacture in every city. We don't want big industries, and after that to send our coatings things. We want to coat everything in each city. That is why small features are very important. And we want to use a small amount of liquids, and also it's obvious we would like to have energy efficient. So, and in this slide, we can see my system that I designed during my master degree. So at the top we can see a camera, and here at the left side we can see a side view camera. I use these cameras in order to in order to see my experiments from left and from the top, and after that I'm analyzing the videos. So what I'm doing during my experiments, I will tell you in a few minutes. But I would like to mention that this part is the surface acoustic actuator that is here. As you can see in my mouse, here we have electrodes, and this is works like USB. I plug in my surface acoustic wave actuator, and I put electrodes, and after that I apply voltage. And when I'm applying the voltage, so surface acoustic wave propagates from right side to the left side. This is in this slide you can see a measurement of surface acoustic wave. We use a Rayleigh type wave. So at the right side there is an image of my measurement. I measured it using laser Doppler vibrometer. So I would like to explain what is surface acoustic wave. Surface acoustic wave is a wave that propagates, in this case, from right to left. And Rayleigh wave is a wave that propagates only on the surface. What I mean, it decays exponentially with depth. And after 3 wave lengths, it almost left only 1%. That means that most of the power propagates on the surface. And we use this power in order to control the liquids and the coatings effects. In this video, we can see how surface acoustic wave spreads under the surface acoustic waves. So the surface acoustic waves propagate from left to right in this video, and you can see that the silicone oil film starts to flow. Sorry, okay. In this slide, we have 2 videos. One from a side view, and the second one is from the upper view. And as you can see on the left side, here there is a silicone oil film that moves under the actuating of the surface acoustic wave and to fully covering the water drop. And here we can see the same from the upper view. Why it's important to fully cover and why we use water drop. Because water drop is spherical and it's well defined. And it's only the beginning. Now we understand the physical phenomenon of coating, which is induced by surface acoustic wave. And from that, we can make every application of coating that is needed. It's important to notice that in these scales, it works in a low gravity environment, and that is why I talked about spacecrafts and all the space issues. So in this slide, there is
Avital, you have 1 minute left.
Thank you. Yeah. It's I have only 2 slides. In this slide, you can see a partially covering. I have a water drop here. This is the top of the water drop, and the oil only covers partially. Why? Because I didn't apply enough voltage, and we understand that we need to apply only a voltage of 1.4 or above in order to achieve a fully covering. So conclusions. So surface acoustic wave may induce liquid coating flows without any using moving parts, and it's great because it's less tend to malfunction. Also, we show that coating of a water drop by an oil film as a model of systems, as I talked before. And after that, we can make new systems. And successful coating is obtained above a power threshold, which is 1.4 volts. And it's important also to make it small and scalable. And in my case, it's applicable at a low gravity. So thank you.
Thank you so much for awesome presentation. Okay, so if you have any questions from the attendees, please go ahead and ask. Okay, Iris, go ahead. Yeah.
Okay, so I have a question. You used the silica oil. So
yes.
If you would use another material, it would work too. Like, what are the parameters that affect if it would work or not? Would it work with anything?
Okay, my surface acoustic wave actuator is made of lithium niobate, piezoelectric lithium niobate. If I worked with different material, I can work, different surface acoustic wave actuator, so I can use different kinds of liquids. But for this liquid, silicone oil spreads over this material, but other materials doesn't spread, like water drop. That is why I use the water drop and the silicone oil. But yes, I can build different systems.
Great, thanks. Any more questions from the audience? Okay, so Avital, you talked about low gravity applications. So yeah. Do you have like a specific application? Because there are in low gravity, you can go, you have many applications there. So you have any specific one in mind, or?
Yeah. And
Yeah, sure.
I prepared some slides for it. Okay.
Yeah, go ahead.
I thought that you would ask, or someone. Okay. Today, in order to code, I need to deep coating. So I need gravity. But when I use surface acoustic waves, if the length is below 1 millimeter in my system, so I can the gravity is negligible compared to the capillary forces. Therefore, at this size, on the left side, you see the drop that is down. What is mean down? If I have my system and I put the drop here on the right side, so on the left side, the video is I just flipped it and it worked the same. I made the analysis and I saw that it's the same moving. So I understand that I can coat like that. If I want to make a coating, I can put a reservoir on the right side and it can be a coating application. But now I also only made the physical principle. I try to understand the physical principle of coating.
Okay.
My answer was yeah.
Yeah. So no, I'm just confused a bit. I mean, yeah, sorry. You're saying that like you can do drop down and drop up coating, right? So
Yeah.
it's like, so just for my knowledge, drop up is like in lower. You're saying it's against gravity?
No, when the drop is up like that, so it's with gravity. It's like against gravity, so I need gravity. But I wanted to check if the gravity has an influence. So
Yeah,
I flipped my experiment, and it was the same flowing, same rate, same coating effects, all the same. And it was amazing.
Okay.
My opinion, yeah.
So okay,
you can see also here the pictures, and you can see it's the same. It's like I freeze the movie. Yeah. Okay.
Thanks. Great work and all the best. Now,
thank you.
Okay, now we'll move on to the other speaker of this session, Hani. Hi. You can share your presentation, and we're happy to hear you.
Sure. Everyone seeing? Hello all.
Yeah, we see your presentation.
Hi, I'm Hani Barhoum from Tel Aviv University, under the supervision of Pavel Ginsburg for Dynamics of Nanostructures lab. We're doing a lot of stuff, but mainly we're lately doing some work around vaterite. So what is vaterite? We'll talk about it. I will talk about it in brief. Everything will be in brief in this talk. So I will confine my talk about in this thing about main things that are done, and especially the functionality and the decoration of this biocompatible micro nanoparticles. So what is the vaterite? It's mesoporous, it's very porous material. It has a very big surface area, and it can be loaded with various molecules, particles, everything that you want. Here's the images of different type of stuff that I put into this porous matrix, like gold nanoparticles. This is a confocal image, and here we can see drug delivery into cancer cells. Here it's it's loaded with biodots, which are nanopeptides, fluorescent material. And we want to do with it different stuff. We want it to be bioimager. We want it to make drug delivery. We want it to be a therapeutic tool. So we want it to multifunctioning because it have high load capacity. So, and a lot of bright contrast in optical region. And this is our main tool. We are optics lab, so we're doing all our measurements are revolving around optics. Here you can see the SEM image of this material, and here we can see it loaded with different particles. You can see nice colors. And it can be loaded with fluorophores. We can load it with quantum dots, with plasmonic particles. Here we can see them inside the matrix in TEM image. Here we can see SEM and TEM. You can see this is magnetic nanoparticles. So what can we do with this thing? Before we start talking about what we can do with this thing, let's talk about what we can how we synthesize it, or how we use the synthesis. So first, we studied its optical properties. This material is anisotropic, meaning it has a tensorial refractive index, and luckily it is pretty bright relative to a very low refractive index material. So we here can see the full tensorial matrix of this material. We simulate this thing and we got this material is also found in some alpine plants, and it can enhance the light collection in plants. And we investigated this thing in our paper, and this is simulations and some dark field measurements which can help us to penetrate the diffraction limit of light, and we can go to the nanoscale imaging of objects. So in another paper, we managed to control the release and loading of materials into this porous vaterite by making different vaterites in different shapes, different size, by simple method of synthesis, like very mild, very simple to synthesize. Here we can see, and it's not only that the shape changes, it's all properties changes, like zeta potential, and when zeta potential changes, the different material can be integrated in and released in the way that we are interested in. So we control the material that we can put in by making layers by layers, or making different shapes or different sizes. Here we can see confocal images of cells cells with particles penetrating through the membrane. The red small dots here, and we can see that different shapes can penetrate the cells in different way, and they have different surface areas, so different loading, different zeta potential, so different also releasing. So this way we can tune the drug delivery. It's very biocompatible. You see the viability. It doesn't change even in very high concentrations. Here we can see confocal images. We can see that in different shapes, we have different loads, different releases rates of this material. Like here, we can see the Langmuir. We can see that toroidal particles have the highest load capacity, while the sphericals have less. And here we can see that the best release, or the fastest release, occurs in elliptical particles. And here the viability tests, we can see like it's pretty negligible. It's very, very biocompatible, so it can be used in research, and it can be used also on humans. So what next we're planning to do with this material? We have a new project, like to have a tuning, like to triggering of the release. This is a confocal image of the particle. This is different areas. This is the particle, and this is the background. So we're using some trigger It's in patent levels, so I can't say exactly what is it, but we're using this. This is in this case we didn't put the trigger here inside the particles, so we see that the particle there's a bleaching of the dye over the particle, and in the background we see almost no changes.
Hani, you have a few seconds left.
Okay, so we have here the particles that were loaded with the trigger, so we can see that the bleaching over the particles, but the background is going up and it's by 14%. So we triggered the release by optical trigger, so it's a nice thing. So this is what we're doing with this material, and we are trying to make with it diagnostics, like taking it into cells and to see what's happening inside. So here we can see it with loaded with some dye in optical tweezers in our lab, and here it's loaded with plasmonic nanoparticles, so we can see that it can generate heat. So it's for heat therapy and something like that. Here we can use it for imaging. We can track the particles and the properties, or track the dynamic medium around the particle to know what the environment, to track the environmental changes in the cell or in the tissue, depending on what. And
Hani, I will have to cut you off. Ah, great. Okay. Okay. So, anyone please have a question? Please ask now. Okay. So, Hani, first of all, am I pronouncing your name correctly?
Yes, you're pronouncing
Okay.
it pretty much good, Hani.
Okay. So, the question is, do you like, I have like a lot of questions. First is, do you yourself make these particles in your
Of
lab?
course, I make the
Okay.
particles, and I make also the particles that are around. I make the plasmonic particles,
Yeah.
the quantum
Mhm.
dots, the carbon dots, only the fluorophores we buy, like not all of them
Okay.
also. Some of them we make in our lab. We're trying to make a biocompatible platform for diagnostics.
Okay. So what technique do you use to make these?
To make the particles, it's like a simple method. I use viscous medium of ethylene glycol instead of water. You can make them in water, but when you use water, the reaction of crystallization occurs very fast, so you can't control the size or the morphology. So we use viscous medium and low concentrations to control the sizes and the particles. So it is done by wet chemistry, totally wet chemistry, but sometimes
Okay.
we do some feed, some advanced stuff if we want to make some certain feature on the particle or some certain stuff.
Okay. So and you make different shapes as you showed.
Yes, we can make them in. We managed to make them in toroidal particles and in spherical and elliptical and different. You see, like we can change the ellipticity, we can change the spherical, we can change the sizes. You see here we have 2 nanometers, 200 nanometers, we have 50, we have 3 micron. We can make them big, we can make them super elliptic, and we can make them also toroidal particles like those with the hole inside. And all of them have different surface properties that can be used.
And the encapsulation of various, what you encapsulate, and they are in the matrix of the system?
The encapsulation, I do it in vitro. I do it outside.
Okay.
I can load any material, then I wash it out. And
like via adsorption? By adsorption? Or, I mean, you incubate the the drug that you want to encapsulate with the system?
Yes. Of course, every drug that you want to encapsulate, you need to functionalize sometimes the surface so it fits the chemical interaction. So it will be loaded in the amounts that you want into the particles.
Yeah, so it's like post synthesis of the particles, right?
It is like what?
Like it's post synthesis part, like after you synthesize the valerite and then you do adsorption.
You can do it also simultaneously.
Okay.
You can trap the particles while synthesis. It's also an option.
And what kind of things that you can encapsulate, like hydrophilic, hydrophobic? What kind of?
You can do hydrophilic. It's easily done, But hydrophobic, it's a bit of a challenge. Depends on the hydrophobicity. So actually, it's one of my main barriers in achieving a good platform in our new project, because my new molecule is very hydrophobic, and I'm trying to find the best solution for it to be loaded and fairly loaded into the matrix.
Okay, thank you so much. Very nice talk, and we'll move on to our next speaker, who is Gal Yosefi. Gal, you can share your screen and let's hear you.
Okay, just a second. Do you see my screen?
Yes.
Yeah. Okay, so you hear me as well. That's good.
Yeah.
Okay. So hi, everyone. My name is Gal Yosefi. I'm a PhD student at Professor Ronit Biton's lab at the Chemical Engineering Department at Ben-Gurion University. My research deals with the formation of macroscopic membranes self-assembled between biopolymers and peptides. So our overall aim is to design and create a new type of platform for in vitro cell culture in the form of a biocompatible macroscopic membrane, which will be formed from peptides and polymers. Our work is inspired by work done by Professor Samuel Stoop almost a decade ago, even a bit more, in which by a multi-component self-assembly, they were able to form membranes composed of positively charged peptide amphiphile. Peptide amphiphiles is a sequence of amino acids connected to an alkyl tail, and a negatively charged biopolymer named hyaluronic acid. These membranes were shown to have 3 distinct regions and were found to be very useful for a variety of applications, for example, cancer therapy. We decided to choose 2 other components to prepare our membranes, an amphiphilic and positively charged PFK peptide, which I will discuss more in the next slide. Our peptide is composed only of amino acid groups without the alkyl tail, and a negatively charged polysaccharide named Alginate. So PFK is a cationic and amphiphilic beta sheet peptide. It is composed of the following sequence. And when we dissolved PFK in water, its molecules self-assemble into fibers, as you can see here in the cryo-TEM image, where the charged side groups point to its outer surface. So we form our membranes by combining both solutions of the alginate and the peptide. And the membranes are formed at the liquid-liquid interface. Now, we wanted to prepare membranes in 2 shapes, a planar membrane and a spherical one. So in order to prepare planar membrane, we just placed one solution on top of the other so that the interface is planar. And in order to prepare spherical membranes, we place a drop of the alginate solution into the peptide solution. So we have a spherical membrane. As you can see, we indeed were able to form these macroscopic membranes. And I should mention that it's not that trivial, because usually when you combine solutions with opposite charges, you will get precipitation. In this case, due to a very unique mechanism, we were able to get these macroscopic membranes. So we were able to form these membranes, and now we wanted to study a bit more about them. And simply by observing them, we saw that there's some kind of a change in their macrostructure over time. As you can see, both in the planar membrane and the spherical one, we have some kind of a thickening over time. So we know that we have some kind of a change in the macro scale, and then we wondered whether or not we will have some kind of a change in the micro and nanoscale. So we took our membranes into to SEM and for SAX. SAX is small angle X-ray scattering. And we saw that eventually all of our membranes can be divided into two main groups, non-fully developed membranes and fully developed membranes. As you can probably guess, fully developed membranes were called that way because eventually we were able to make all of our membranes to have this kind of structure. So let's start with the non-fully developed membranes. You can see it has like some kind of an amorphous zone, and on top the contact layer. In the SAX curve, which is intensity as a function of the scattering vector, we have a single peak. This peak represents the contribution of the peptide fiber diameter. In the fully developed membrane, we see that we have additional fibers that emerge from the contact layer of the membrane. These fibers are perpendicular to the contact layer, and we can see them not only in the SEM, but also in the SAX. So in the SAX curve, we see that we have two additional peaks. These peaks represent the contribution from the distances between these fibers. So now we know we can form macroscopic membranes, and they have a very unique and cool structure. But we wanted to see if we can determine ahead of time which structure we will get when we prepare membranes. And due to the fact that our membranes are different than the ones published before, we had to identify a mechanism for their formation. So due to lack of time, I'm not gonna describe the whole mechanism, but I will explain a bit about the steps that are necessary in order to form a fully developed membranes. So first, we have a rapid formation of a mixing barrier, followed by the formation of a semi-permeable membrane. So actually, these first two steps are controlled by a reaction diffusion process. And the non-fully developed membranes I showed you the slide before are actually membranes that didn't pass the second step. The third step is achieving a sufficient osmotic pressure difference, resulting with the formation of these perpendicular fibers. And this step is controlled by the osmotic pressure. So now we know what steps we need in order to form a fully developed membranes. But now we wanted to see if we can make our membranes and specifically control the structure that we will get. So we were able to pinpoint the key parameters that affect the membrane structure. The first one was the membrane incubation time. As you can see, the comparison between a 1-day incubation and 6 days of incubation. Peptide aging period, you can see the membrane composed of fresh peptide compared to aged peptide. Membrane shape, you can see planar membrane and a spherical membrane, and the peptide and polymer concentrations. So here's the membrane composed of 2-weight% of alginate, and here a 3-weight% of alginate. So actually we were able to find exactly the parameters that can specifically control the structure of our membrane, enabling us to use these membrane for a wide range of applications. And these results were recently published. And if you will want to read more about it, you're welcome. Today, we're working on two main things. The first thing is to examine the biocompatibility of these membranes. Here you can see a cryosem image we took of a cell on top of our membrane. The second thing we're doing is designing and creating new types of membranes composed of different variety of biopolymers. So I want to say thank you for everyone part of this project, and thank you for listening.
Thank you so much. Very nice talk. And if we have questions from the audience, you can go ahead and ask.
I have a question. I wanted to ask about the beta structure of this material and how does it form?
So we have the molecules of the peptide, and you can see here. So we have like two molecules of peptide combined together on the side.
How did you resolve the structure, the molecular structure? How did you resolve it?
We did FTIL.
How did you resolve the molecular structure?
Oh, this image?
Yes.
Actually, it's not my image. It's an image. The collaboration we have is the one that did this image. So I'm not sure which program they used.
So they make it in molecular dynamics.
Yeah, sure.
Yeah, nice. And yeah, very interesting, the beta structures, because they have a lot of some of them, like we had also a publication about some fluorescent, like if you do, they become from alpha to beta, they become fluorescent, and it's like a very expanding field.
Okay, if we have more questions. Okay, so can you move to your slide number 9? Just have some doubt. So you said that the formation of semi-permeable membrane is a very important step, right?
Mm-hmm.
So so I mean, why is it so? Can you elaborate on that?
Yeah, sure. So actually, it's quite interesting because this step is unique to our membranes compared to previously published ones. And the reason is that we saw that even though we can start, we have two main things that must happen. The first one is a mixing barrier. If not, we will have a chaotic mixing and a precipitation. The second thing is we need a very large differences in the osmotic pressure. So we saw that even though we can start with a very large difference in the osmotic pressure and the mixing barrier forms, we still don't always have a fully developed membrane. But if we will wait enough time and enough of a complexation interaction will occur, then a fully developed membrane will occur. And we understood that the middle step that we are missing is the fact that we need some kind of a semi-permeable membrane to form in order for the osmotic pressure difference to enhanced.
Okay, so this step is possible only if you give it enough time to develop, and it all depends on the time, or there are other parameters.
No, it doesn't only depend on the time. There are some parameters that are affected by time, the membrane incubation time and the peptide aging period.
Okay.
But for example, the membrane shape, so it's quite interesting, but both of these membranes are formed by fresh peptide and membrane that were incubated only 1 day. So it's like very short time. And you can see that spherical membranes already have these perpendicular fibers. So they are fully developed in very, very short time.
Okay. Okay, so I guess that's all. Thank you so much for awesome presentations, and I guess we have a little bit of break, and then we have a plenary session 2, and I hope you all will be there because it's gonna be awesome. So thanks again, and see you next year. Okay.
Thank you.
Bye-bye. Take care. Bye-bye.
Bye.
Thank you.